Okay, hi. It's great to see so many people here. I hope you didn't stay just because you have a seat now in this room. But instead, I'm assuming that you're either interested in FAIR principles or in protocol biases in microbiome sequencing data. And that means that we share at least one research interest. So, hi, I'm Luise Rauer. I'm a doctoral researcher at the University of Augsburg and the Technical University of Munich in Germany. And I'm presenting our meta-analysis of bacterial mock communities. So whenever we do microbiome sequencing, our data are basically distorted by multiple biases that we introduce in the sample processing. So basically, we can introduce biases during the sampling, during the DNA extraction, during amplification and sequencing, and even our bioinformatic and statistical choices affect the results that we get in the end. So basically, our observed composition of the microbiome almost never represents what we expected in the beginning. How do I know the expected composition? Well, I'm working with mock communities. So mock communities are samples with a known composition and known species properties. So we know exactly what we put in, and then we can also measure what we get out. And there are cell mock communities where we have just bacteria, and DNA mock communities where we have DNA of the bacteria. And the good thing is that many people use these mock communities as positive controls, for example, in clinical microbiome studies. And I'm interested in these protocol biases. So I thought, hey, if people are using these positive controls, let's just exploit the vast variety of protocols that is already out there. And let's do a meta-analysis of these mock communities. So this is what we did. So first, we identified commercial bacterial mock communities. There are around 34 from 3 different companies. Then we searched for publications using these mock communities in Google Scholar. And then we applied inclusion and exclusion criteria. And this is the full scheme. I would like to highlight 2 things here. So first, we focused on Illumina 16S sequencing, which was basically just the most widely used method. So we were able to keep 79% of our studies. But then in contrast, the major exclusion criterion was that actually the raw data was not available. So here we were only able to keep around 30% of our studies. And I would like to show you this in a little more detail. So here we have the number of studies that we included in our analysis and the year when they were published. So you can see microbiome publications are nicely going up. Now, what I expected in terms of publications depositing their data or not depositing their data is that it goes down, right? Journals are increasingly enforcing these rules that you should deposit your data, but it's actually going up in the same way. And the main of course, so sometimes you find this available upon request, which in principle is fine. But then there were also cases when the information was stated where to find the raw data, but then there is actually no raw data. So here's one example from 2018. I mean, we are all busy as scientists, but I don't think that these researchers are still working on uploading their data. And for the last exclusion criterion, I would like to play a little game with you, which I call Spot the Mock. So, so here I have all the raw data that was uploaded in the study. And the only information that we have is blah, 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 incorporation of a mock community during sequencing. Who knows what could be the mock sample in this case? Yeah, it's quite hard. So I think in this case, it was the PC positive control, which makes sense. But there's also a lot of other abbreviations here, and there's no additional information. And this was actually one of the easier cases. So this is a study that we included in the end. So this last category, again, just has the same rate. It's also going up. So we just have a significant proportion of articles that either do not deposit their raw data or where the raw data is just not clearly labeled. Okay, so now we have applied our inclusion and exclusion criteria. Then we were downloading the FASTQ files, processing them jointly to generate a huge ASV table, which just contains the samples and then basically the genera, and then like count data. And then from the publications, we extracted the sample metadata, so how these samples were actually generated. And this brings us to the problem. Basically, how do we squeeze these tons of different pipelines that are available for generating microbiome data into one scheme? And luckily, there's already some work done. So maybe you are aware of the STORMS checklist or of the MICS specifications. So building on these publications, we collected around 100 variables of these sample metadata from the publications. And then we can analyze how frequently this metadata is reported in the publications. So here on the left side, we have just the publications that use the CellMock community. So it includes all the steps that you apply to a CellMock community. And on the right side, we have all the steps that come after DNA extraction so that you would also apply to a DNA mock community. And you can see that, for example, the extraction kit or the hypervariable region, like the really key information of any microbiome project, they are given in 100% of the cases. But then other information, for example, on the PCR conditions, like how many PCR cycles did you use, there the information already goes drastically down to 2/3 of the study. When it comes to the second cycle of PCR, we have only 1/3 of the study reporting the details. So there's some information lacking here. And this is what the data looks like in the end. So we have 4 different mock communities shown in this figure. And I think the most important thing that you can see is that there's quite some variability, first within a mock community, so just from the different protocols, but also even within a study, there's quite huge variability, which is basically just random variability. But you can also quite easily distinguish the mock communities from each other, which is a good message for us, right? It means if you have totally complete samples in your microbiome experiment, you process them with totally different pipelines, you still get totally separated samples. That's a good sign, right? But we were not interested in finding differences between mock communities, but in finding differences between the protocols. So basically, we corrected for the expected composition, focused only on the shared genera, removed some outliers, and now the mock communities are nicely mixed, and we can start studying some biases. Unfortunately, or fortunately, we see some differences based on the hypervariable region, and I'm sure that there would be more differences, for example, based on the primers that were used in the studies. But in this analysis, we are really limited first by the information that is given in the papers, as you have seen before. And on the other hand, we are limited by the fact that some of these studies are so specific that we cannot really say if that's just a study-specific effect or if that's really because of the methodology. However, when we look at the cell mocks, it looks a little better. So here we can clearly see that we have huge effects based on the extraction kits, and we can explain up to 93% of the variation for some of the genera just based on the extraction kit that was used. If you're interested in extraction bias, I'm also here with another poster, which I present tonight in the evening. It's number 235. And yeah, we can bioinformatically correct extraction bias. So I'm happy to also discuss this if you're further interested. But basically, to summarize this project, we first found that there's basically really lacking data availability in terms of the data that has been deposited, and there's no improvement over time at the moment. Second, there's also missing, or that we miss the critical sample metadata in order for us to do the study, but also that would be needed for this full laboratory reproducibility. But in general, we have this powerful approach where we can take the mock communities and measure all these biases. We are limited at the moment, basically, in the step where we are extracting the sample metadata from the publication. So if anyone has an idea how to automatically extract this content from the publications, I would be happy to hear about that. And to give you a few more general conclusions, I would be happy if you use mock communities as positive controls in your research. This would also help you to see if your results are really working. And I would like to say that I'm really not here to judge anyone. I found mistakes in my own research like this, where I was not adhering to these standards. But if you can, please try to adhere to the reporting standards and try to go fair. And with that, I would like to thank my co-authors, and I would like to thank you for your attention, and I'm happy to take questions.